I hope you still have some energy for another 15 minutes or 20 minutes. So today I'll tell you something about revisiting our thinking on cancer genomic or cancer predisposition. And thanks for the previous talk that really put all what I need for this one. So very quickly, I'll say a few words about evolution, cancer evolution. Then I'll introduce our method called VarI, in which we seek selection, evolutionary selection for cancer sample. Then we'll move to population, and I'll explain what we do with cancer predisposition within a population, in this case, and then few words about ultra rare private mutation and what can we do with them. And if time allows, where are we heading? So cancer evolution. This very nice scheme just tell us what cells are doing when they divide and start to take their own fate. And what I like in this scheme is that you can convert it to classical evolutionary tree, like phylogenetic tree, and that's exactly what we see in those, let's say, two cases in which they are metastatic 1, metastatic 2, and so on. And why is it so important? We just heard about driver genes, and of course, driver genes are those that really drive the progression of the cancer, while they are hidden within a bunch of passenger genes that are sometimes as important, but we would like to look at those that drive the story. So just to give an example that we just heard about, P53, a very well-known cancer genes, this is the length of the protein, and you can see just counting how many mutation in each spot, and you can easily see that there are some hotspots. And indeed, for example, in this position, there is arginine that was changed to histidine with thousands of reports as of now. And you can see that this is very informative. This has come from cosmic data and so on. However, I call this method frequentist view because we count. So as a counter, we know that this is an important driver gene. However, we should mention that those are prone to a lot of mistakes or some kind of biases. We heard about few, but I want to add, it's not only the hotspot, but the length of the gene and how much CG and what's the signature and how much DNA repair and so on and so forth. So we want to overcome those genuine problems. And indeed, this is what we are trying to do. The notion is very simple. We say that all genes playing role, namely driver genes in cancer, are expected to show sign of positive selection. And this is exactly the same slide that we have shown, in which what we want to look for is the difference or the access of specific mutation over the neutral one. And why is it so important? Because the cancer, from the point of view of the cancer, there is some optimization to any of the hallmarks of cancer, which is, for example, angiogenesis or genomic instability and so on. So positive selection is working towards this. And that's exactly what we want to capture. So how we do it? Okay, good point to stop. So you were patient with me to wait for my sentence, okay? I can keep talking?
Yes.
Yeah, sure. Okay, no problem. So the idea of positive selection is really crucial, and we wanted to change the frequentist view to a functionalist view. What does it mean? So we understand what each and every mutation is doing to function. Which function? Protein function. So we take the easy way. So we're just trying to understand what happened to the protein itself. And for that, we had to build up some kind of machine learning, not too complicated, but in which we included thousands of features, as you can imagine, not mysterious features, and to build and train for ClinVar or whatever we had, as we just heard, and to make something that we can go on with. So I'll be quick now. So the idea is to find protein or genes that are more damaged than expected. I need to define what's damage and what's more damage than expected, what's expected. So for that, we need a machine, as I told you, that will transform variant to the impact on biochemistry of the protein. And that's exactly what we did. And this is the FIRM. FIRM is a functional inference of mutation in which we cooked thousands of features and we trained them to CleanVar, and we have this trained model in which FIRM score is defined by capturing the likelihood of a gene to retain, let's say, 20% or 50% of his original function. So it's a relative probabilistic type of score from 0 to 1. And then we move to the data of TCGA. In this case, we only use the DNA sequence and not the other excellent resources that are there for different projects. And we collected out of the 3.2 million variants within the exome, the 2.2 that are within the coding region of the exome. And that's our resource. So now we are going back to 101 in biochemistry, not even 101, 100. So the effect size of the observed versus the expected, what that it means. This is the genetic code. And we take the cancer from TCGA, and we ask, let's say, the mutation in P53 that I just showed, what will be the outcome from the table, from this genetic code, and what firms are telling us about the effect size. And we do the same for the background, but there the background is based on the 12 transition A to C, A to G that we have, and we do the same. What will the genetic table will tell us and what firm will tell us, and then we just compare. If we have significant difference, we said, okay, now we know that this is significant. So let me elaborate a little bit more about this part, the background, because I think this is the most important part. What we do here, we take a sequence, and whenever, let's say I'm just looking at A to G, just one out of the 12 transition that I mentioned. So A to G, every time that I see A, I change to G. And I ask, what happened? And then I ask firm, what do you say? And I get a distribution. And this distribution will be compared to the distribution of the observed, let's say 53 and all the other genes that I collected within the cancer. And that's I do for the 12 cases, but I need to have another small correction. This is 80 million calculations, so on. But the correction is that A to G in this specific gene will be different than G to A, for example. In this case, it's 3% versus 25%. So I have to correct each gene. I don't ask why. I'm not asking why it's so different, but I'm using it to correct the matrices, to multiply those matrices. And this is the end of fabric. We have observation, we have background that I explained pretty deeply, and then we have the effect size, yes or no, statistically. When we did this, we took the TCGA and we found 600 genes. Quite interestingly, 400 of them were known. Known means either in the database of driver genes or in a paper that claim, but about 200 were new. So what are they? Why they were missed or not missed? So this is where we started to investigate, and I'm just showing you one slide in which we took the 5 most, let's say, significant finding from the unknown, overlooked genes, and we looked into different data sets, so it will be completely independent and so on. And when we did this, we did a survival curve, and we saw that those 5 genes, indeed, mutation within these 5 genes will affect the survival curve of this population from 18 months to 26 months, but significantly. So there is something there in this set, and that we, just as an example for what we do with this 200. So fabric as of now, I told you about pan-cancer, as if cancer is one disease, but we know that it's not. So we repeated this, but now on a specific cancer type. And when we did this, we also went back to all those 33 cancer types, and we also looked for which of those driver genes or cancer genes, let's call them, will be also unique for a specific one, because that's more important for the clinical point of view. And indeed, this is the list. As you can see, in the top of the list is P53, the well-known. But as you can see, also, if we do another z-value for the uniqueness, now P53 is in the bottom, because it's general. It won't help the clinician if they look for specific, let's say, therapy or so. Another genes that were in the middle of their way went to be up in this list. So this is what we do. And as many of our approaches end up with a database and a website, Fabric is a website, you can enter whatever you want, cancer, gene, and so on, and get the results. So once we have this idea, I want to move into what we do with population. That was somatic. So this is the PWAS. As of now, I told you about somatic, and I mentioned those two databases, the TCGA and the MSK. But now I want to move back to the population, to the germline, the predisposition. And the predisposition must use either population or family. And I'll show you example of each very briefly. So for that, we use the UK Biobank and the GNOMED and so on. And the definition of gene predisposition is quite obvious, so I don't have time to define it, but I just say that 40 years of work, really hard work, got to a very narrow list of predisposition. I just show you a few very famous ones, the BRCA1, BRCA2, and so on, but the list is limited. So why is that? We went to the UK Biobank, which is really a great resource, with genomics, but also medical record, lifestyle, and so on and so forth. And we ask the same idea that I mentioned before. Instead of jumping from the variant to the phenotype, in this case, cancer, we want to do it through the protein understanding. How? I told you, through FERM. So that's what we did. But before showing you the result, I want to tell you something that is very trivial, but I want to emphasize. When people are doing GWAS, usually they do 1, 2, 3 to capture this scheme. However, we added another layer, which is called recessive, in which we wanted to ask at the level of the gene whether we have cases in which a mutation in two alleles are different position are still affecting only as a combination. So that's so I call it R for recessive. And we use, now we take the UK Biobank, let's say any cancer, and we take FERM and we ask what FERM tell us about the recessive model and the dominant model. So two model for each gene, 20,000 gene. And then we do something very, very naive. Case control, isn't there any difference from statistics? That's all on the distribution, as I showed you before. And when we do this is one example of this. We started with 5,000 people. We filter them out to avoid family members and all this. We ended up with this number, and 20% of them did have some cancer, which is quite a lot. Now we focus on 10 cancer, and we were able to find from classical GWAS about 900 variant, and from PWAS that I just mentioned, this is the protein-based WAS, we found 100 gene, association gene. We combined them to make sure that we are not overlapping. Altogether, we found 137 loci. Quite interestingly, only 10% were shared between cancer, which is completely different from what I showed you in the somatic. So now each cancer, it's its own signature. That's another surprise that is worth to mention. Another surprise was that never mind if we take, now this is the 137, if we take GWAS or only PWAS, our method, or those that were discovered by two method, we see that a substantial fraction has no evidence, so they are new. Why is that? Second surprise for you? It's because we are capturing recessive. So what you see in this color is the PWAS discovery, in which 2/3 or 60% are because of recessive. That was completely missed by GWAS and so on. So this is another kind of revisiting cancer. This is the reason for this title. So the last few minutes really is on ultra rare and private mutation. We use PWAS also as a website with 30 different cancer-related, and this is just an example with 500 cases of bile duct and liver disease, a cancer, in which we did what I told you before, and this is the result. This is what was significantly surprising, and when you look at the list, don't try to understand the list, but just look at the color. The reddish color are those that increase the risk in cancer. The blue one are those that are protective gene. And we can see that in this case, we also are able to find nicely protective genes that usually are ignored as well. Is it just one case? So we did it for all cancers, and this is just a sample. You can see that in some cases, you have 40% of the gene discovered that are protective. In some other cases, it's only, so it's a case by case, but it was important for me to mention that recessive, not only recessive and protective are two elements that often are not considered seriously enough. To finish, I'll skip this. To finish, just to say that recently we used family. In Israel, we have very unique family. First of all, big families. And second, that the families are kind of, we know generation after generation, so you can pick up the DNA, which is quite unique, I would say, in Israel. And we made up some kind of a artificial AI-based ranking for exome, and we were able to discuss 12 families with about 40 exome, full exome, and to try to find whether we find predisposition in this specific case. We found very nice and interesting, also protective and risk genes, and we found that they are very much related to the origin of the family. So that was all in different, non-dependent family of Persian origin and so on. So we were able to find ultra rare cases very nicely. What next? I mentioned UK Biobank, but there is a tsunami of other biobanks, really interesting, really good. We are using some of them now. And the takeaway, I started with FIRM as a method for scoring. I implemented it in somatic and in germline. I show you the family power of private ethnic-based CPG, predisposition gene. And I jumped over some PWAS core in which we expand the germline into genetic discovery by ultra-rare variant. I'll stop here, but I cannot stop before saying that we are living in a very, very tough time, and it has to be ended. And I mean, I cannot have a day without thinking about it. Too much suffer in the Middle East to everybody. And thanks for you and to my great group.
Yes. Great talk. Thank you very much, Michal. I probably didn't understand something, but when in the first part, in the molecular part, when you do the frequentist approach and you count the mutations, As you know, certain genes are a lot more studied than other genes. P53, BRCA are studied a lot more.
Absolutely.
So how do you avoid selection bias? Meaning, when you get to the background, you would know that P53, you have a lot more association with cancer than, say, some Joe Schmo gene that nobody really looks at that much.
Excellent. So what we do is something even simpler than you think. We take the number of variants that were picked, let's say in P53, thousands. And we did the background with the same number as observed. That's it. So if in this Cucuricu gene that you just mentioned, there are only 100, we do just 100. And probably...
Because Cucuricu can have more than 100, right?
Yeah, but
just weren't studied.
Still, we do 100 couple of times just to make sure that we are not biased by numbers. So we overcome this.
Understood.
It's a good comment.
That was awesome. Can you hear me?
Yeah.
Awesome. Why not synonymous variants?
Why not?
Synonymous variants.
Okay. So as of now, our, let's say, PWAS or firm is very naive, saying if you have a stop codon, it's 0. You kill the protein. We know that it's nonsense, because if you have a stop codon in the last amino acid, who cares, right? And the same idea was for synonymous, unless it's on the splicing side. On the splicing side, we took it very seriously, and actually we did some cleverness around the splicing, but all the rest is a little bit naive to say that it keeps 100% of the functionality. We have some work showing the effect on translation rate, but I still think it's minor relative to the, but it has to be, I mean, it will be nice to refine, let's say, the firm. Now we are refining it with rare, ultra rare variants, and we already see that it was hidden. A lot of information was hidden that we were not aware. So probably we are going to the easy part, like all of us.
We actually see synonymous variants in cancer. So
yeah, we can talk about it. I think it's super important.
I think your tool would work really well for them. Why not? It seems like it's a...
We include them, but we call them one. We include them, but we give them the best score. We include them. We are not, yeah.
All right, for the interest of time, let's move to the last speaker today. Thanks, Michal, again.